a technique by which a fluorescent antibody was made to bind to the antigen immobilized in a tissue section, so that one could detect the position of specific protein molecules in cells by their fluorescence. And shown here is a modern image. This is a nuclear body from a salamander oocyte. Here it shows immunostaining with the protein, with an antibody against the protein cortin. Here's immunostaining with an antibody against the SM protein. This is a slide which I made more recently using the Bunsen-Kaplan technique. The point, however, is that they had shown that it was possible to detect specific protein molecules in tissues. And it occurred to me that it would be wonderful if one could detect specific nucleic acids in tissues. Actually, I was thinking less about nucleic acids as I was thinking about genes, because I've been studying the giant chromosomes of salamanders shown here, or the giant chromosomes of Drosophila shown here. And in these chromosomes, one can see bands that were pretty much regarded as genes by people working with them at that time. We now know it's a little more complicated than that, but it seemed that we had here a tissue, a chromosome, where specific genes, specific nucleic acids, it should be possible to do something comparable to the immunofluorescent staining technique to detect specific genes in these tissues. So actually, I kept this in my mind for quite a long time, and it became possible to think about doing this kind of thing in the early 1960s, when investigators discovered that one could take double-stranded DNA, denature it, into a single strand, and immobilize those strands on the nitrocellulose filter. This was done by a couple of investigators named Gillespie and Spiegelman. And what they showed was that having the denatured DNA on a filter, one could then hybridize radioactive RNA to it and actually determine quantitatively the amount of a particular gene sequence on the filter. So I thought, well, if they can do it on the filter, you ought to be able to do it on a slide. So I tried denaturing the DNA on a slide that is in cells, nuclei that were squashed out onto a slide, and I found that I could denature the DNA quite adequately without damaging the tissue. But then when I tried to hybridize a radioactive RNA to it, I got no signal. In this case, I was looking for the radioactivity by autoradiography, that is, by covering the tissue with a thin layer of photographic emulsion and looking for silver grains in the emulsion caused by the radioactivity of the probe. As I said, nothing happened, and I more or less gave up on that technique. That was in the early to mid-1960s. Now, a break came quite accidentally from studies that I and others have been carrying out on salamander oocytes, frog oocytes. And what we found was that in the giant oocyte nucleus, which is shown here, derived from this large oocyte here, the oocyte is the egg of the frog, inside of this giant nucleus are, of course, the chromosomes, but in addition, we found that there were several hundred to 1,000 nucleoli, these round bodies here shown on this particular slide. Now, the interesting thing about these nucleoli that we discovered was that they contained DNA. And it was specifically the genes that code for ribosomal RNA that were in these nuclei. This was a most unusual situation to have DNA outside of the chromosomes. It was quite clear from work that had been carried out in my laboratory, as well as in the laboratory of John Brown and of Oscar Miller, that there was extrachromosomal DNA outside in these nuclei. And I studied the origin of that DNA, and what I found was that it actually was synthesized at one specific time in the early history of the formation of the oocyte. And one could see in these early cells, early nuclei, that there was a cap of DNA here, and you can see the cap over here. You can see the chromosomes here, and down here you can see the chromosomes in this nucleus. So in these two nuclei, you have the chromosome, but then you have this huge cap of extra DNA, which in fact was the ribosomal genes that had been somehow come out of the chromosome and amplified to give you this one specific sequence, a very large amount of this one specific sequence. So it occurred to me that if there was any possibility of doing in situ hybridization, this was the case that would work. And so I took the radioactive ribosomal RNA and hybridized it to cells of this sort, and I was quite pleased to see in the very first experiments that we got good in situ hybridization. Here, this cell is black because of the silver grains in the photographic emulsion that have been exposed by the radioactivity of the hybridizing probe. So this is a case where we have hybridized radioactive RNA, ribosomal RNA, to the ribosomal DNA in the cell. So that led immediately to a publication with my student, Mary Lou Pardue, who joined me in this work. And this was the first successful in situ hybridization. Now, we wanted to try it on some other tissue or DNA, but there were very few examples because at that time it was not possible to isolate specific DNA RNA sequences. What permitted us to formulation came from studies that had been carried out on mouse DNA. And investigators have found that when mouse DNA was spun in the cesium-chloride gradient, it separated into two fractions, a main peak and a satellite peak, called satellite simply because it had a slightly different density and separated from the bulk of the DNA. We took this satellite DNA, made it radioactive, and we hybridized it to mouse chromosomes, not knowing at all whether it would hybridize, whether it was even in the chromosomes for that matter. It might have been a virus or some other infecting particle. But we were very pleased to see that the mouse satellite DNA hybridized specifically to one portion of each chromosome. And you can see on this slide that each of the chromosomes has a bit of radioactivity at the very end of the chromosome. This region of the chromosome was known as the heterochromatin and had been known cytologically as a specific region of the chromosome for a number of years. This led us immediately to think that we should be able to try the same technique and get similar results looking at the chromosomes of Drosophila. And the chromosomes of Drosophila were especially exciting because it was known from genetics that the genes were all in the so-called euchromatic regions of the chromosome, namely this region from here to here, or over here, these lighter stained regions. All of the genes were located in those regions, and it was known from genetics that the heterochromatic regions, like here and here, were devoid of genes. So it occurred to us that Drosophila must have a lot of DNA similar to the satellite DNA of the mouse. So we centrifuged that DNA in the cesium-chloride gradient and found indeed that Drosophila had a lot of satellite DNA. This species of Drosophila virilis that we were working on had three huge satellite peaks here, here, and here, in addition to the main peak of DNA shown here. So we took the satellite DNA, we made it radioactive, we hybridized it to the chromosomes, and sure enough, it hybridized as shown on these images here to the heterochromatic regions. And this was particularly interesting, as I said, because it showed that the heterochromatic regions that we knew from genetics had no genes consisted of this satellite-type DNA, which our later studies showed consisted of very simple sequences which were incapable of coding for genes, or they were capable of being genes and coding for proteins. So the in situ hybridization technique at this point was limited pretty much to sequences that were present in large amounts in cells. Eventually, it became possible by having more and more sensitive probes, and particularly later techniques were worked out for making fluorescence probes to do in situ hybridization. So in situ hybridization, ISH or ISH became fluorescence in situ hybridization, or FISH, F-I-S-H, which is the way it's usually practiced now. And techniques have gotten more and more sensitive to the point where nowadays it's possible to detect individual single DNA and RNA molecules in tissue sections.
My name is Elizabeth Blackburn, and I'm going to tell you about the discovery of telomeric DNA and of telomerase. When I first started working on this question, the endochromosomes, these are chromosomes here, were known to be very important for protecting the genetic material, but nobody knew what was there. And it was known that every chromosome had a long DNA molecule that goes from one end of the chromosome to the other, and that long DNA, of course, carries all the genes. And every time that chromosome is replicated, the DNA has to be fully copied. But it was also known around the 1970s that the DNA replication machinery of the cell wasn't able to completely copy all the way out to the very edge of the chromosome. And so the prediction of what might happen every time the DNA replicated in order for the cell to divide would look something like what's depicted in this slide here. This would be that the chromosomal DNA end region would get shorter and shorter and shorter until eventually the cells would have such short chromosomes that they would be perhaps missing something from their ends and then not able to divide anymore. And that was even predicted on these theoretical grounds to be something that's called senescence, which had been seen in cells growing in concert, that they, in certain cases, could only go through a certain number of divisions and then they couldn't divide any further. But nobody knew why. And so, how to approach this problem? Well, I began it by taking advantage of a particular kind of organism, which is called Tetrahymena thermophila, and it's a single cell ciliated protozoan. And you can see in the scanning electron micrograph here, all the cilia all over the single cell. And the particular reason for this choice of organism was that these cells have within them some very short linear chromosomes in high numbers. And so what was known before was that while nobody knew what was at the ends of the chromosomes in eukaryotes, cells with nuclei such as Tetrahymena, certain very short linear viruses, which called bacteriophages, had the ends of their DNA analyzed. And in this organism, Tetrahymena, the very short chromosomes were about the same kind of size, and so I thought, well, I would try and look and see what is at the ends of these chromosomes, these mini chromosomes. Now, at this point, DNA sequencing methods had not been developed. And so, although this is hard to agree probably these days, but that was the case. And so what I had to do was to rely on a different kind of approach, and that was to really try to stitch together pieces of information about the ends of the DNA that I could obtain by using labeling techniques, radio labeling techniques, which would incorporate radio labels into the DNA, and analyzing by combinations of enzymes and chemicals that would free the DNA into little pieces that were dependent upon a particular building block basis of the DNA, and patch together the sequences, so stitch it together like a jigsaw puzzle. So, for example, I would cut the DNA up and see that there were, for example, little motifs in it, like this one here, where there was this motif of 4 cytosine residues in a row, CCCC, that's C4. And in fact, furthermore, I could tell from some of these analyses that there was a purine, that is an A or a G, and then CCCC, and then an A or a G, because of the way I chemically cleaved the DNA up. And furthermore, by quantifying what was there, I could find that there were actually many pieces of this little motif at the ends of these short mini chromosomes. And so, I put it all together, I was able to conclude that these little tiny chromosomes ended in something that had really never been expected, and that was a sequence that formed the strand, which is the complement of the CCCC strand, it's actually CCCCC, and it's just repeated over and over and over again, perhaps 20 to 50 different repeats. Now the question is, how do these repeats get there? And at this point, there were a lot of possibilities that you could draw on paper. And a lot of the times what you do is you take an observation and you put it into the existing body of information. You say, well, how can I explain minute hiding given the context of all the information we know? So we knew about DNA, we knew about its replication machinery, we knew how it would be combined. And there were certain well-worked-out rules by then for this. But these kinds of DNA sequences were not obeying those rules. And I'll tell you how they were obeying those rules in a moment. But the first thing I think that's interesting is that there comes a point when you're working in science where you know a lot of things, you're trying to fit your results into well-established principles, and then it comes to the point where it just won't fit in that box anymore, it just can't push into that box anymore, and you have to say, well, let's entertain some other possibilities. Now, the kinds of observations I won't go through them were that there were variable numbers of repeats on these small linear chromosomes, sometimes there were 30 or so, sometimes there were 50, 70, there were different numbers of repeats. Well, that was already odd, didn't look like the bacterial viruses. And then there were some other observations, one of which came from the extraordinary biology of these particular organisms, so we set behind the Thermophila as an example. The way it's got very small chromosomes is because it has a stage in its development right after fertilization when it chops its chromosomes up into small pieces, and then these telomeric CTG4 repeats would appear at the end. How did they get there? This was not obeying rules that were known for DNA. DNA was supposed to be copied from DNA, or at least recombined with DNA that was very similar. You weren't supposed to have two sequences suddenly joined together. And where did these repeats come from anyway? So I decided to look in extracts of the tetrahymena cells and see if one could detect if there was some enzymatic activity that might be able to add this sequence, TTTTT, over and over again, to the ends of linear DNAs. I didn't know what kind of assay really to use at this point. But as you know, there's a stage in the life of these organisms when the very short chromosomes got created. And in fact, they were created by chopping up the DNA, for stories which they were created by chopping up the DNA for reasons that are a whole different story. And then the telomeric, as we call them, repeat sequences, were being somehow put onto the ends of these DNAs. Often in places where there were no such sequences there before. So how does this different sequence get joined onto another piece of DNA? That is what we were trying to think about answering the question, how did these repeats get put up? And so, after a lot of trying different assay conditions, I finally found a mixture that was able to give me the first hint that something was happening, because I could see increasing amounts of this sequence being synthesized apparently from nothing in the test tube. And then at that point, I was joined by a new student in my lab, Carol Brandon. And so the challenge now was to simplify down this reaction that we had going on in order to be able to see if there really was an enzymatic activity that truly was doing what these experiments had been hinting at. And so Carol was able to refine them, to strip down the assay to its bare essentials, to get to the following point, the picture here. So we would make a DNA oligonucleotide, which is this colored bar here, made up of the building blocks you can see that look like the end of a chromosomal DNA. And then, by adding the extract of tetrahymena cells right at the stage when they were known to be making new telomeres, because the chromosomes at that point in their life cycle were being chopped up and telomeres were being added. So I reasoned that they might be enriched for any such enzymatic activity if such a thing existed. So they made extracts from cells right at the stage and found just by adding simple salts and two simple building blocks of DNA, GGP and TGP, that in fact repeats were added to the ends of these DNA chromosome end mimics. So this is very exciting for us because they said, this really would be a potential way of solving this problem of DNA getting shorter and shorter, because now here's a way of making DNA get longer. So we had an enzyme activity which was working in the test tube and adding nucleotides that corresponded to the telomeric DNA sequences with Solomon, and then ACE sounds like an enzyme, and we were thinking of polymerase, where, a polymerase is something that makes a polymer. So we said, well, here's telomerase that makes a telomere. So we were happy to add this new word, which eventually made its way into the dictionary. Now, just to give you an example of the kind of way these reactions look, what this is depicting here is an autoradiogram, which is a fractionated mixture of the reaction products, which have been labeled with trace amounts of radioactivity, and then they've been fractionated in an electrophoresis system, what's called DNA sequencing cell. And they start with just a very short DNA oligonucleotide, we call it the primer, and then the DNA is longer and longer and longer. And you can see that there's this lovely repeating pattern, and that pattern is 6-phase repeats with the TT motif being repeated over and over. And you can see that with time, more and more of this gets added to the ends of chromosomes. So this is a very visual kind of demonstration of this activity, and we could quantify it and do many experiments to try and understand its nature. One of the things we found out not too long after first finding this activity was that the enzyme actually had within it an essential ribonucleic acid component. And what this does is depicted here. So it had a ribonucleic acid component in it, which made the enzyme very sensitive to the enzyme ribonuclease, not what you'd expect if it was just a protein enzyme. And within this RNA, there was a sequence shown in blue here, which is the exact complement of the TTGGG sequence that we found was being synthesized in the test tube. So with such a very strong hit, we decided this really was the enzyme, and now the challenge was to find out if this really was behaving in the cells as it was behaving in the test tube. Because what we were finding in the test tube was that the DNA, such as mimic of the chromosomal end shown in black here, could be elongated by addition of nucleotides and copying the template, which was our model, and that would now make the chromosomal end longer. Was that really happening in cells? So the answer to this gave us a bonus answer as well. We were answering this question by making small changes in that blue sequence here, in the RNA, and we're putting the RNAs with the change sequence into the cell and asking if the change sequence in the blue sequence caused a change in the DNA that was added. For example, if this templating mechanism, as it's called, were really true, then if we changed a particular C into a G, then instead of being copied by templated synthesis into the G, it would now be copied to the C. Or for example, if we changed one of those A's into a G, it would now be copied into a C instead of into its normal T. And in fact, that was what we were able to show in experiments that were done in cells by Guolian Yu, John Bradley, and Lara Atadi. Now that also gave us very unexpectedly another answer to this question. How do tetrahymetal cells respond when telomerase is not working? We weren't actually initially setting out to do this experiment, but serendipitously, one of those changes in the template sequence, or in fact a couple of different ones, gave the sorts of effects that are shown here. So certain changes, for reasons to this day we really don't understand, would cause the enzyme, instead of simply synthesizing the corresponding repeated sequence with the corresponding mutation in the DNA that was copied from the template, instead, these changes caused the enzyme to choke up, and it wouldn't work. It just basically stopped the enzyme working. So it just sort of got choked up in its active site and can't work properly. And that was really lucky for us, because it allowed us to say, what happens when an enzyme that doesn't work is present in the cell? And what happened was that this telomeres started getting shorter and shorter, and then over the course of about 20 to 25 cell divisions, they progressively got shorter and shorter, and then the cells ceased to divide all together. And so that told us something very important, if we can sort of summarize here. Tetrahymetal cells are normally immortal. That is to say, they keep multiplying pretty much forever. However, all we had to do was to inactivate telomerase by this very small surgical strap in the central RNA component of the enzyme, which inactivated the enzyme. And now the cells became, if you will, mortal. They could have a certain number of cell divisions, during which their telomeres progressively shortened, they got too short, and then the cells ceased to divide. So, loss of functional telomerase is leading to the complete loss of telomeric DNA from the chromosomal end, just as had been predicted from the original predictions about DNA replication with some history mechanism. And so, many cells have sufficient telomerase, and so they can now maintain telomeres at various lengths, but maintain sufficiently long so cells can keep multiplying. When we discovered telomerase, we really by trying to answer a very basic question, which is how do chromosomal DNA solve the problem of incomplete replication? And it was very much driven by, first of all, the idea that if there's some interesting problem in biology you want to go for it and try and understand how nature is working. I think in the back of my head I was always aware that nature tends to be very conserved in many of its most fundamental molecular mechanisms, as has been amply learned from the central dogma of DNA information going to RNA, going to protein. Although interestingly, this is a case in which a perfectly normal cellular enzyme was breaking the rule and RNA was being copied into DNA. Something that people before had been only the kind of things that certain viruses that